In this video, we're going to address the fact, we'll talk a little bit about the DNA polymerase enzyme and the fact that it is not perfect. So first of all, here is a protein diagram of the enzyme itself. So you can see here, this thing here. It's a giant enzyme. It is a very complicated protein. It's a quaternary protein, which means there are multiple polypeptide chains or subunits kind of glued together to build this big enzyme. You can see the different colors. There's probably about 6 or 7, 8 different pieces that are jigsaw puzzled together to create this enormous enzyme. You can sort of see where the active site is right here. This is where the DNA strand sits and where it matches up the bases, the A to the C, the A to the T and the G to the C. That's where the magic happens. But you can also see on this diagram something down here called the exonuclease domain. So this is another part of the DNA polymerase III enzyme. Now, you can see at the top that the title of this slide is that replication is not perfect, that DNA polymerase makes errors. It also corrects most of the errors that it makes, but it doesn't catch them all. Now, what do I mean when I say it makes errors? Well, it's cruising down the template strand that it's reading, and if there's an A, it's going to stick a T nucleotide for the new strand. And if there's a G on the existing template, it's going to put a C for the new one it's building. And if it sees a C down here, it's going to put a G up here. So it builds its new strand, the new strand that it's building, based on the instructions that it's seeing from the existing strand. So it's kind of like somebody typing really quickly, putting letters in, G, A, T, T, A, C, G, A, whatever it sees, it's building the opposite, all right, or the pair up. But every now and then, it puts the wrong base in. So if it sees an A and it's supposed to put a T, but it accidentally grabs a G and it puts a G there. All right, so it's made a typo or a mistake. Now the enzyme keeps going a little bit, but that exonuclease, so you can imagine it, the enzyme moving along. It's also got a little bit hanging out its butt that kind of feels the piece of DNA that it just made, and it says, does that feel right? Is it lining up right? Is the bases pairs correctly? And if it can feel that the bases aren't lining up right, like there's an A and it's trying to pair with a G, which it shouldn't be, the DNA polymerase enzyme can sense that or feel that, and it can stop moving forward, and it can backtrack and pull out the bases that it put in wrong until it gets to the error. So it pulls out the G, and then it puts a T in there, which it's supposed to be, and then off it goes again. So it's basically like when you type and you see a typo, and then you go backspace, backspace, backspace to get to where the error is, take out the error, and then type on correctly. All right, so DNA polymerase does put in mistakes. It can generally go back and fix them, but it doesn't catch every single mistake it makes. And it leaves a typo roughly on average around 1 in a billion letters. 1 in a billion. If you could type that well, you'd be like the best human typist that ever was, right? So it's very, very accurate, but it still leaves a mistake 1 in a billion. Well, how significant is 1 in a billion? Well, in a human cell, one of your cells, the entire 46 chromosomes in a single cell have about 6 billion nucleotides strung together to make 46 individual chromosomes. So a total of 6 billion nucleotides. So when an individual human cell divides into 2, all 6 billion nucleotides need to be copied into new chromosomes to make new chromosomes for the new cell. And if this enzyme is leaving an error 1 in a billion, how many errors are going to be there? Well, on average, about 6 errors every time the DNA is copied. All right, and remember, the cell only copies its DNA once in its life when it's getting ready to divide. So this doesn't happen very often necessarily, but I want you to think back to, well, you probably can't think back this far, but there was a time when you were 1 cell big. When you were conceived, when an egg and a sperm came together, so however old you are plus 9 more months, you were conceived and you were 1 cell big. And that 1 cell has divided and then divided and divided and divided, 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 divided many, many times to build your entire body. All right, so let's go back to when you were 1 cell big, so however old plus 9 extra months, right? When that 1 cell divided into 2, now there's 6 errors in the new cell. When those 2 cells divide into 4, now there's another 6 errors, so that's 12. And when those 4 divide into 8, there's another 6 errors, so that's 18. Now remember, this is just on average, so it's not going to be 6 exactly every time. But imagine how many times your DNA has been copied over your lifetime to go from 1 cell to the 13 trillion cells that you are right now, and that it's still getting copied. Like every day you need to make new skin, and you need to build more liver cells, you need to build this, that, and the other. Every time those cells the DNA is being copied to build new cells, errors are creeping in. Now, clearly, it's not fatal, right? You're fine. You've got errors in your DNA compared to when you conceived. Apparently, no big deal, because you can deal with it, right? There's reasons why it's not fatal. We'll talk about that in the next topic, the next week. But the errors do add up over time, and we gain more and more of them the longer and longer that we live, and the more our DNA is copied. And oftentimes, cancer is a disease that can be caused by errors from DNA being copied. And we tend to think of cancer as being a disease as you age, a disease of more of the elderly. Now, I'm not saying there aren't situations where cancer doesn't occur in younger people. And we'll talk a little bit about how that happens too in some later videos. But in general, the accumulation of these errors happens more and more and more as you age. And once you get a certain number of them, or specific ones, or areas of your genes get hit by these changes, it's more likelihood that it's going to trigger something like a cancer. Now, I almost said the word there. Whenever you change the DNA sequence by altering the letter order, the A's, the G's, the C's, and T's, that's called a mutation. All right, whenever the DNA letters are changed. So basically, we all carry many mutations. You can't avoid it. And these errors are just happening because of how the DNA polymerase enzyme works, because it leaves an error 1 in a billion. And that's interesting and important to remember, because we tend to always want to have reasons for what caused something to go wrong, right? What made this person get cancer? Something must have happened to them. Something must have been inflicted on them. They must have done something bad to put some bad thing in their body or whatever to cause these errors. Well, there are things that can cause errors, and I'll talk about that in the next video, but there are errors that happen that you can't avoid. This DNA polymerase enzyme is going to put errors in your DNA regardless of what you do. It's an inbuilt thing that our body can tolerate and puts up with, because there are some potential benefits to it as well. So this is one of the ways mutations happen. These are called spontaneous or natural mutations, because they're just happening because of the DNA polymerase 3 function. You can't avoid it. You're always going to get it, just how the enzyme works. So here's a sort of very quick example of somebody. Let's say this person 1, their DNA goes AAATTT. And this DNA sequence is basically instructions for putting together a protein. And this protein, well, if you follow these instructions, the protein comes out looking normal, which is this kind of shape here. This is what they're saying is the normal protein. All right. This person, when they copied their DNA, they accidentally put a T here instead of an A. However, when their DNA is used, when the instructions are read to make a protein, it still comes out looking like this. So they're going to be totally fine because the protein looks good. Even though their DNA sequence got altered, the protein is still okay. The protein that's made from that DNA is okay. So they wouldn't even know they had a change or a mutation in their DNA. However, this person, that T is changed to a C in this example. So sorry, the A is turned to the C. And when that happens, looks like it really impacts how the protein gets put together. It's all kind of misshapen. And in this case, maybe it doesn't work right, so it's going to cause some kind of clinical physiological issue. Could be very minor, could make your hair blonde instead of brown, or it could be very major. It could give you something like sickle cell anemia, or more highly likely to end up with some disease. We'll address this later on in next week's topic, but these are all mutations. They can be happened by just the DNA polymerase III naturally doing its job. Some of them are fine, some of them cause issues. And why those issues come about, we'll address later.